Good morning, everybody. And good afternoon. I'm going to give a few minutes to give people a chance to sort of come on in. This discussion is going to be on the Mesolimbic pathway, specifically G proteins, and drug intoxication and withdrawals. So we went through the series we went through the mesocortical pathway, and now we're going through the mesolimbic pathway, and we went through the different receptors on these pathways. And one of the receptors are the G protein receptors. This is a family of very sophisticated molecular machines, and they do a lot of the work. They do the work at the molecular level. These G proteins are amazing structures, just fascinating. And they basically, whatever objects a molecule binds to them, it translates the purpose of that molecule to the cell body and does something. So it's going to be a really interesting discussion about G proteins, intoxication, and drug withdrawals. And we're going to look at this in the context of the mesolimbic pathway, since the mesolimbic pathway is the pathway for reward and learning, reward, motivation, and learning. And so this is often the target for substances of abuse, particularly cocaine, methamphetamines, like we went through PCP, LSD, ecstasy, things like that. So hopefully we'll have a really good discussion real soon. And I will be opening a walk-in clinic in the first floor of Jackson Park Hospital on the Stony Island side. It's not open yet, but hopefully in the next couple of weeks we'll have that open. And hopefully I can get it running 7 days a week from 8 a.m. to 8 p.m. So for my patients, and if you need any outpatient services, then hopefully we'll be able to have those services available for you also. The second issue, I'll be opening my online clinic. And so for my patients, if you need to get in touch with me, you can simply download my application, my platform, and just be able to get in direct contact with me without a middleman. So soon I'll be launching my online platform that'll allow me to manage all of my patients and their issues online. At that point, when after I launch my online clinic, then I'll be able to take care of my patients from home, and we'll be able to do all the evaluation from home and give you all the information up front, meaning your diagnosis, your medication, your treatment plan, and things like that. We'll give you all those things up front, and then you can decide what hospital you want to go to. So if it's not a life-threatening emergency, if you don't need to go to the emergency department, then after I open my online clinic, I should be able to evaluate you from home and help you select the hospital you want and try to help you get there to that. We'll help you get there to the hospital. So we're going to have a lot of changes, a lot of new changes and innovations that we're going to be introducing to the healthcare space, and I think it's going to make huge changes. So, for example, I feel the need as a physician to try to give my patients additional tools to help them maintain their autonomy while they're in hospitals. So I'm very concerned about the state of hospitals and the condition that I often see my patients. So we'll be introducing some of those tools into the marketplace and everything, and I hope you like it. So here, okay, for now, let me just go over, let's start now. We can go over G proteins, okay? So let me see here. Let's see here. Okay, so I will use the search engine. Show me how G proteins work. Okay, G protein coupled receptors, also known as 7 transmembrane domain receptors, are a large protein family that acts as signal transducers. And so the last time I read this, it was like 6 transmembranes, so apparently 7. So these are huge proteins that actually go the entire length of the phospholipid bilayer. So it extends the entire length of the phospholipid bilayer. And these components typically work together as subunits. After there's some type of binding of the ligand at one end, it results in a 3D conformational change, and that results in other things. So these things are fascinating. But anyway, anything that binds to them on the outside of the cell, the signal gets transduced on the inside, translated on the inside. Then there's something, a product that's usually created. Um, they are involved in various biological processes, responding to a diverse stimuli like hormones, neurotransmitters, and light. So these G proteins can receive a lot of different type of inputs: hormones, neurotransmitter, even light. Um, Here's how it works: activation. G proteins possess a specific binding site, right? Called for a ligand. When the ligand, such as a hormone, binds to the site, it induces a conformational change in the receptor. This triggers the G protein to bind. This triggers G proteins to bind to the activated receptors. Okay. So next, G protein activation. The G protein, a heterometrics, meaning there's multiple different types of subunits. The G protein is made of different types of subunits, I think 7, and each one of them are different. Composed of alpha, beta, gamma subunits, also associated with the G, is associated in the protein when it's in active state. Okay, when the G protein is activated, the G protein binds it, causes the alpha subunit to release GDP and bind GDP. Okay, something like that. Great. The exchange of GDP for GTP activates the G protein, causes it to dissociate into two parts. So it causes it to release the alpha and beta subunits. Okay. Signal transduction. Both the activated subunit and the beta gamma dimer can then interact with various intracellular signaling molecules, such as enzymes or ion channels, initiating downstream signaling cascades. Okay. These cascades usually lead to a specific cellular response, like a gene expression. Right? Terminating the signal. To terminate the signal, the alpha subunit hydrolyze GDP to GTP. GTP to GDP again, causing it to reassociate it with the beta gamma dimer, inactivating G protein. Makes sense. Okay, so that's sort of like the sort of like framework of how it works. Ligand binds, conformational changes, then it produces stuff that produces stuff from the cell body, or produces stuff that it exports from the cell. And so remember, on the mesolimbic pathway, on this mesolimbic pathway, it has NMDA receptors and glutamate receptors, and these glutamate receptors are G proteins, and so this is how they work. Just look at the next section here. Let me see if I can answer a question here. Okay, so if you have any questions, please feel free to sort of let me know, and I'll sort of stop as I go along. Okay, so first question for T-Jam. Hey, T-Jam, thanks for the question, T-Jam. Why do some people feel euphoria while others feel paranoia from intoxication? The euphoria would be activation of that mesolimbic system, right? And paranoia, activation of that mesocortical symptom, because you get activation of the mesocortical pathway, you get positive symptoms. Some of the positive symptoms could be perceptual disturbances, paranoia, things like that. Or, these drugs also impact the ego. The ego can also cause you to sort of feel a sense of paranoia. These are two different networks. Thanks for the question. Next question from Muhammad. Thanks, Muhammad, for the question. Is intoxication purely a dopamine effect, or do G proteins play a separate role? Intoxication is associated with the mesolimbic pathway, which is a highly dopaminergic pathway. And these dopamine receptor, and that the pathway has G proteins, right? Glutamate is a G protein, right? So glutamate is on the pathway. If you get over excitation of glutamate, you get hyperactivity of the pathway, and the glutamate receptor is a G protein. Okay. Okay. Let me use the next question. Next section here. I wanted to, let's see, explain ion channel depolarization secondary to explain ion channel depolarization secondary to G protein activation and substance dependency. Explain ion channel depolarization secondary to G protein activation and substance abuse dependency. Okay. In substance abuse, the intoxication is not mediated through the G protein. It's mediated through the chloride channels that's in close proximity to it. And the intoxication is secondary to the degree of depolarization. All right. So here, my question was explain how the chloride channel next to the G protein, it affects intoxication and withdrawal. Okay, here's a question. Depolarization of ion channels, okay, secondary to G protein activation. Okay, so the G protein is activated, and then you get depolarization of the ion channel. It plays a significant role in substance dependency by influencing neuronal excitability and synaptic plasticity. Right. And so this opening and closing of chloride channel affects the excitability of the neuron and also the learning part. That's the plasticity. Here, how does this process unfold? Okay, neurotransmitters binding to the G protein activation. Okay, addictive substances or neurotransmitters like dopamine bind to specific G protein coupled receptors on the neuron membrane. Okay. This binding triggers a conformational change in the G protein, causing it to activate the associated heterometric G protein. Okay. The activated G protein promotes the exchange of GDP to GTP and the alpha subunit, leading to the dissociation of alpha subunit, okay, from the dimer. Okay. We went through that on the previous discussion when the alpha and the beta subunit separates from the G protein, and that's the activated state. Okay, next, G protein mediated modulation of the ion channels. Okay. It does it by direct gating or by second messenger pathways. Okay. In some cases, the activated G protein subunits can bind directly to and gate ion channels, all influencing the opening and closing. Oh, wow. So the subunits, the activated subunits themselves can bind to the G protein channels. The degree of depolarization of the channel it gives you an indication of the degree of intoxication, right? And and over time, the channel becomes less sensitive to the G proteins, so you don't get as much depolarization. And therefore, you may have to use a substance for longer or something like that. Second, it also does it by direct gating by the G proteins themselves, directly interacting with the ion gate channels. Second, the second messenger system. Okay, more commonly, G proteins activate intracellular signaling pathways, include involving second messenger systems like cyclic AMP. Okay, and we'll go through this a little. I'll go through this on the diagram. Okay. Impact of neuronal activity and synaptic plasticity. Okay. Excitatory postsynaptic potentials. This depolarization, if sufficient, can reach the threshold for generating an action potential, transmitting a signal down the neuron. This is sort of like the system, the level never goes back to baseline. So it takes, you only have to do the difference in order to spark an action potential. Synaptic plasticity. This G protein mediated depolarization, along with other downstream signal cascades, can lead to long lasting changes in synaptic strength, known as synaptic plasticity, which is like long-term potentiation. Okay. And then here, long-term potentiation addiction. In the context of substance dependency, this process is particularly relevant in reward pathways in the brain. Psychostimulus, for example, induce long-term potentiation at an excitatory synapse on dopamine neurons in the VTA. Okay, activating dopamine receptors. Okay, this strengthens the synapses in the reward system, and it contributes to the reinforcing properties of drugs and the formation of drug-seeking behaviors. Okay, I hope that wasn't too complicated. Let's see here. Okay, I have another question here. Okay, let's see here from Muhammad. Let's see here. Is it possible to block or alter G protein signaling to reduce intoxication effect? Okay, so we're talking about the dopamine receptor, and I'm not sure if we're going to block the dopamine receptor. I'm not sure. I'm not sure of that question. Um, I don't. Okay, is it possible to block or alter G protein signature? I don't. I don't think it is. To answer that question, I don't think so. Let's see. Okay, next. Okay, Hannah. Okay, thanks for the question, Hannah. Doc, are G protein effects reversible after long term drug use? Um, we're going to get to that a little bit later. That this chronic use results in basically down regulation of some of the receptors, and because you down regulate the receptors, then when you no longer take the drug, the system isn't able to sort of balance itself. And so we'll get to that a little bit later. So hopefully I'll be able to answer that question. Okay, thank you so much. Okay, here, let me get to this next section here. Okay, here's a diagram that I found here. This is the a basic cell, just a basic cell here. Phospholipid bilayer. Here's a G protein transcending the phospholipid bilayer. Then it has this alpha, gamma, beta supplex, things like this, right? Okay. Now notice, let me see, how they have here. They have this leading to this, and I think they have this leading to this, and did they have this leading to this? I don't know. Okay. But anyway, so here's the molecule attaching here and making resulting in 3D conformational change. And the conformational, and the result of the ligand binding to the outside of the G protein results in changes on the inside. It's going to either secrete a substance here from the cell during this process, right? Or is going to send instructions to the cell body to create a product, or is going to open a channel. So you look at the complexity of these molecules, these molecular machines, the G proteins, and the range of molecules that they can attach to create conformational changes, to create instructions for the cell body and coordinate the response of what's happening to the cell body. It's amazing to see. And all of these diverse proteins either result in three things: secretion of a product out of the cell, instructions for the cell, the nucleus, to create a product, or open or closing channels. Here. Explain cocaine withdrawal at the molecular level, including the receptors. So we're talking about the mesocortical pathway, highly dopaminergic, so it's impacted in cocaine, methamphetamines, ecstasy use, things like that. Also, the mesolimbic pathway is a target for cocaine, and we see the cocaine toxicity As our patients with cocaine dependency issues typically have very severe memory problems, and we pick those up on our neuropsych testing. So let's see here. Okay. When someone becomes dependent on cocaine and stops using it, they experience cocaine withdrawals, a complex neurobiological process involving significant changes at the molecular level, and cellular level in various brain regions revolved in reward, motivation, and stress, right? Mesocortical, mesolimbic, mesocortical. Initial mechanism of action of cocaine. Cocaine's euphoric and rewarding effects are primarily due to its actions on the brain reward pathway, right? Mesocortical, particularly in the nucleus accumbens. I think that's where that mesocortical begins, somewhere around there, right? Cocaine achieves this by blocking the reuptake of neurotransmitters like dopamine. On the presynaptic cell are reuptake pumps. The purpose of these reuptake pumps is to pull these substances that you're secreting, ejecting out of the cell back into the cell, because you're going to need to build up another supply to start the next round that's going to come out. And then inside the synaptic clefts are enzymes that degrade the molecules. And so you have huge reuptake pumps that always pull the dopamine back into the cell. And so cocaine blocks the uptake of the activity of the reuptake pumps. And so as that presynaptic nerve is sending out the vesicles full of dopamine, there's nothing else coming back in it. And so it eventually runs out of dopamine. And therefore that synaptic cleft becomes depleted of dopamine. And therefore you're in dopamine, the whole network is hypoactive, and therefore you're having dopamine withdrawals. So here, cocaine, so it blocks, right, reuptake of dopamine, norepinephrine, serotonin, right, by their respective transporters, the reuptake transporters. Okay. Leading to an accumulation of these neurotransmitters in the synaptic cleft. That's cocaine. The surge in dopamine levels in particular activates dopamine 1 and 2 on the postsynaptic neurons of the nuclear accumbens, which contributes to the feeling of pleasure and reward. Great, we got that mechanism of action. So basically it blocks all the reuptake pumps and all the neurotransmitters build up in the space. Neuroadaptations in chronic cocaine use. Okay. With prolonged cocaine use, the brain adapts to the heightened dopamine levels by reducing its natural dopamine production and decreasing the number of dopamine receptors in the reward pathway. Doesn't need that much. These are the compensatory mechanisms to normalize the excessive dopamine signaling. Wow. Wow. So with chronic cocaine use, you get down regulation of the dopamine receptors. Yeah. This process of regulation is known as tolerance, where the individual requires higher doses of cocaine to receive the desired effect. Yep, you got me. So hit the reuptake pumps. That's easy. Turning the reuptake pumps is pretty easy. Just turn them off. And then chronic use, start to download receptors. Chronic cocaine use can also induce changes in glutamate transmission. Okay, yeah, I knew glutamate was going to be here, some story, because we see excessive agitation, combative aggressiveness, particularly during cocaine intoxication, which involves alterations in AMPA and NMDA receptors in the brain, like the nuclear accumbens and prefrontal cortex. And so basically, then the chronic cocaine use alters glutaminergic transmission, and I bet you it alters it for the worse. I think it makes it more active, and I think that's why you can get the agitation, combativeness, and all that stuff. In the nuclear accumbens, withdrawal from repeated cocaine exposure has been shown to result in increased levels of brain-derived neurotropic factors, which lead to selective downregulation of certain glutamate receptors, impairing the induction of some type of long-term depression. Oh wow. Okay. There is evidence suggesting that possible downregulation. There is evidence suggesting that possible downregulation of D2 receptors functional response via interactions between Okay. Molecular mechanisms during cocaine withdrawal. When cocaine is stopped, the brain's adapted state is disrupted, leading to a sudden and significant dopamine deficit, which contributes to the withdrawal symptoms, including depression, anxiety, and anhedonia. Remember, you blocked all the reuptake of serotonin, norepinephrine, and dopamine, and then the cocaine caused the release of those in the synaptic cleft. And I guess all of it was metabolized or left the region through volume transmission, diffusion. Okay, here, so when it's stopped, okay, it's disrupted. This dopamine deficit and the reduced sensitivity of the reward system make it difficult for individuals to experience pleasure from its natural rewards, reinforcing the cycle of addiction, making relapse more relapse. So if you run out of all of those neurotransmitter in the presynaptic cell, at some point, you're going to have a absence of them in the synaptic space. If it's not there, then the whole transmission on the mesocortical pathway is hypoactive. And if the pathway is hypoactive, then you get the cluster of symptoms associated with mesolimbic pathway hypoactivity. And that's what we see with drug and cocaine withdrawal when we see the patients in the morning. And this I don't want to get into any else that are here. Let me just do here. I just looked up methamphetamines. Real quick. Okay, let me just see if there's anything different. Methamphetamine withdrawal symptoms are profoundly influenced by G protein changes, okay, signaling pathways in the brain, particularly those involving neurotransmitter. Okay, methamphetamines significantly elevated extracellular dopamine level by increasing release and inhibit uptake. Okay, so methamphetamines does the same pathway. Chronic use leads to damage of the dopamine pathway, damage of the dopamine system, potentially affecting both dopamine level and the number of dopamine transporter and receptors. Yeah, so this is the same thing. The chronic changes. They didn't say what impact the chronic cocaine changes has on the reuptake pumps. They just mentioned how much the chronic cocaine use have on the impact of the postsynaptic receptors, and it so causes down regulation of those receptors. But I wonder what chronic change is happening to those reuptake pumps. This damage disrupts the delicate balance between dopamine transition, leads to a downward balance of state of imbalance upon sensation. Okay. G protein coupled receptor withdrawal. Dopamine receptors, which belong to G protein families, are crucial. Okay, D1 receptors activate G proteins and stimulate, increases cycle AMP, which inhibits and D2 decreases cycle AMP. Okay, great. And let me just look. Okay, that's it. Okay, I think that's it for now. So I think I'm going to end the series on the mesolimbic pathway, because I think mesolimbic and the mesocortical pathway, because I think in this series I went through each one of the pathways and went through receptors on each one of them. We're going through the drugs of abuse, the mechanism for each one of them. And I think each video is around at least 20 minutes, probably at the max. I think this is the longest one. So I think I'm going to be finished with this series of this series on the mesolimbic and mesocortical pathway. I don't think I need to review that anymore. Does anyone have any other questions before we end for the day? Just let me know. Okay. So if there are no questions, let me see. Okay, so at the end of this, I want to just reiterate some of the things we have coming up. For all my patients, I'm going to be launching my online clinic, and I would want you to register or sign up on my online clinic so we can stay in direct contact. I want to get to the point where I can evaluate you at home via video conferencing or telepsychiatry and do your entire workup there. And after I got your entire workup there, then I can sort of help you get to the hospital that you want to get to. So I think I'm going to start offering that service for all of my patients to try to get them to the hospital they need. Also, I'll be opening a walk-in clinic at Jackson Park Hospital. That's going to become maybe in the next couple of weeks. So I'm looking forward to that. And so if you're interested in working with me or me taking care of you or someone in your family, then you can just contact me and let me know. Okay. Otherwise, I'll call it a day. Take care. It's a pleasure with everyone. I hope this was helpful for you, everybody. Take care, guys.